So far we have covered the ultrastructure of the cell organelles, and the genetic material, that is nucleic acids, and their mode of replication. And in yesterday's class, we discussed about the cell division, types of cell divisions, etc., and also extranuclear chromosome. So now today, in today's class, we will shift to what we call the another important topic, that is genetics. You know, the term genetics is not new to you. You know, you heard about it, and you know a little bit about the meaning of genetics. So genetics is nothing but study of genes. Study of structure and function of the genes is largely the definition of genetics, or genetics is a branch of biology that deals with the heredity and variation. Now, what do you mean by heredity? Heredity is nothing but transmission of characters from parents to offsprings. That is what we call heredity. And as you know, we resemble our parents because we received genetic material from our father's side and also from our mother's side. So that's why we share many features with our parents and also with our grandparents. So now this is about the genetics. And as you know, genetics is almost more than 100 years old as such, more than 100 years. And of course, before the popularizing experiments that were carried out by Mendel, whom we consider as father of genetics, you know. But before Mendel, you know, many people in those days, of course, it was very, fascinating to think about the origin of various organisms that are present on the earth. And a number of people proposed a number of hypotheses, or they had their own way of viewing the things. Some people before 17th century, people believed in what we call origin of spontaneous origin. Spontaneous origin means organisms arise suddenly in the present form itself. Suddenly they come onto the earth. It is a mystery. Through mysterious phenomenon, organisms do take birth on this planet Earth. This was the feeling of people. This we call spontaneous creation. Spontaneous means suddenly. Nobody knows the reason behind the origin of a given species. So this we call spontaneous origin. But of course, this they have argued in favor of spontaneous origin by giving an example that, say, if you expose either the bread or food, or for example, in Western countries, generally, they take meat. When the meat was exposed, for 2 or 3 days outside, they found small maggots, small germs, small, insects coming from the stored meat. They said that when the meat they have observed, or when they have taken from the animals, it was free from any maggot. Any insect was not there. It was not observed. But where from this maggots came? They said that. So they got into this in a spontaneous way, sudden way. So, but later on, number of people like Pasteur, Tyndall, Louis Pasteur and Tyndall, they disapproved the theory of spontaneous creation. They said that the maggots were observed, or maggots originated in the flesh or the mutton, because it was exposed to environment. In the environment, there are many insects. So insects come and lay their eggs. And these eggs in 2 or 3 days, they hatch into worms. So origin of worms, it was not spontaneous. It was because of the laying of eggs of the various insects, including houseflies. They laid their eggs, and in due course, they hatched into these minute maggots. This is how, and they have proved beyond doubt that, when they covered, they have taken the meat or other food, and they covered foolproof. They did not allow any insect to sit on it. So it was fresh as such. So with this, they have proved that the organisms do not arise spontaneously. All organisms shall arise from their pre-existing parents. So later on, of course, as the time went on in the 18th century, when people or scientists understood the phenomenon of reproduction, marriage, and the egg from the mother and the sperm from the father, etc. And they proposed that, they said that the sperms or the eggs, they contain the figure or the material from the father or from the mother, and it is preserved in that small teeny egg or the sperm in a miniature form. So an individual is there in the sperm itself, or a female parent or mother is there in the miniature form, in the egg. This was the theory of what we call, epigenesis or miniature form. Later on, but of course, as our knowledge of, microscope or cell organelles, etc., more and more important structures and functions were elucidated by the deep study of a number of people across the world. And it was another man, Charles Darwin. Charles Darwin, who is famous for his theory of evolution. He said that origin of organisms. He made it clear in his book Origin of Species, which he published in 1859. In that, he proposes that it is only because of evolution new organisms arise. And he said that he believed that from each body part of an individual, for example, we have eyes, ears, nose, hands, etc. From all organisms, small factors, he called them as gemmules or pangenes. So from each organ, a small pangene comes, and these pangenes pass through the bloodstream and they reach the sex organs. So there they are preserved, and through gametogeny, through the fusion of gametes only, new organism or what we call the zygote formation takes place, and this zygote gives rise to embryo and of course an individual. So he said that he proposed what we call the pangene theory. And later on, of course, it was the concrete evidence for the reason of resemblance, or the scientific reason about heredity was because of our instrumental works done by famous cytologists like Sutton and Boveri. They concluded that the chromosomes are the vehicles. Chromosomes are the vehicles that carry hereditary particles from one generation to other generation. The movement or the behavior of the chromosomes during the cell division. Of course, we learned about it yesterday in mitosis and meiosis. So it is the chromosome. Chromosome is the vehicle that carries the genes, and they are responsible for the transmission of characters from one generation to other generation. A strong scientific foundation for genetics, of course, for our greater understanding of this heredity was because of the works of Sutton and Boveri. And of course, this is in the background, but of course, we now study the genetics proper or the experiments performed by Mendel, and the Mendel theories, Mendel's work, and his principles together we call Mendelism. So Mendelism deals with works of Mendel, his monohybrid ratio, by dihybrid, homozygous, heterozygous, his phenotype, his genotype, all these, and his ratio, all these ratios we call Mendelism. So as you know, Mendel was a he was a preacher. He was a preacher, and he worked in a church in Austria. And because of his curiosity with natural sciences, especially biological sciences, he continued doing a number of experiments, number of breeding experiments in his garden. He was a preacher, and in his leisure time, in the church garden, he observed different types of plants that were there, and he concentrated on the garden pea or Pisum sativum, what we call. And in the Pisum sativum, and why he has chosen Pisum sativum, because large number of varieties of Pisum sativum were available to him in the garden. Lot of variants, lot of differences he was able to see in the Pisum sativum, and he observed, he selected, 7 pairs of characters. He selected 7 pairs of characters. Say, for example, a tall plant, a dwarf or a short plant, a plant that produced rounded seeds, a plant that produced wrinkled seeds, a plant pea, Pisum sativum only, garden pea, which has a smooth seed coat and wrinkled seed coat, and the cotyledons which were yellow, cotyledons which were green, and the flowers that were axillary flowers that were terminal, and the part that was without any constriction, the other with constrictions, and the parts color, etc., etc. In all, he has selected 7 pairs of characters. So he has selected like this 7 different varieties of Pisum sativum having distinct characters, and these they had alternates. For example, one, if you take tall, its opposite one is dwarf, round, wrinkled, yellow, green, axial, terminal. So like this, he has he has chosen 7 pairs, and he has taken, he had a good knowledge of math. He was good at mathematics. He was brilliant. He had a brilliant knowledge of mathematics and also statistics, and he used this knowledge of mathematics and statistics to make permutations and combinations, calculations, and based on that, he was able to publish his works, his hybridization results in Natural History Congress. So this a brief background. Now coming to, as you know, because briefly we will be knowing. See, as you know, tall, to avoid confusion, he has used letters to denote a particular character. Tall means he has given, he has taken capital T. For dwarf, he has taken small t. Round, or color, for example, yellow, Y, green, GR, G, green. So like this, he has given, capital letters for the characters that appeared in the F1, that is first generation. Say, for example, he has taken a tall and a dwarf, and these two are differing in how many characters? At a time, he has chosen only one character. The character is length of the stem. Character is the length of the stem. So based on the length of the stem, he was able to have 2 different types of plants. One was tall, the other was short. And these plants, which are differing in one character only, and he is crossing 2 parents, which are differing in only one gene or one character. And he called such crosses as monohybrid crosses. Say, for example, tall it was represented by what we call capital T, capital T. And dwarf was represented by small t, small t. When he crossed a tall and a dwarf, he got an hybrid, and he named that hybrid as F1, first filial generation. And this F1 parent or the F1 hybrid was tall only. F1 hybrid was tall. And he's subjected this F1 for self-pollination, selfing, this we call. So in hybridization, what he has done, he has taken a female, one plant as a female parent, the other plant as a male parent. From the female parent or from the flower of the female flower, he removed the stamens and he has dusted the pollen from the dwarf flower. So like this, the characters of both the parents were mixed. And whatever the F1 generation he got, it showed or it exhibited tallness. And when in the second generation, he did not excise or remove the stamens. Emasculation was not done. He allowed the plant because the flowers in Pisum sativum are, bisexual. It has both pistil and stamens. So it was because of that, selfing was possible, and the transfer of the pollen of the same flower was onto the stigma of the same flower. So this we call self-pollination or selfing, and it resulted in the second generation. And to his surprise, in the second generation, he was able to find not only tall plants, but also dwarf plants or short plants. Now, what happened to the dwarf character in the F1 generation? Now, he proposed a hypothesis, which is called a principle or a law of dominance. Law of dominance means the character that appears in the first filial generation and the character that gets subsided or that gets hidden and reappears in the F2 generation is known as a recessive. So dominant character and a recessive feature. Dominant feature and a recessive feature. So, what I mean to say here is dominant character was expressed in the F1 generation and the recessive character reappeared in the second generation only. F2 he called. F1 means first filial. F2 means second filial generation. Now, the plant, the phenotype, what we call, how the organism looks like, that we call phenotype. And based on its genetics or genes, gene characteristics, we call genotype. And external features we call phenotype. So phenotypically, first generation all tall. Second generation, tallness and dwarfness were in the ratio of 3:1. 3 were tall and 1 was dwarf. So this we call, and he proposed the law of dominance. Law of dominance says that only of the two alternates, and these alternates genes are called what we call nowadays, we call them alleles. So one allele is expressed, the other allele gets hidden and it reappears in the second generation. So the allele that appears, or the character that appears in the F1 generation is called dominant. So dominant and recessive appeared in what we call 3:1 ratio. Now, he proposed, see, the dwarf character is present in the F1 generation, but though it is present in F1, it is not getting mixed up, no blending, no mixtures. What is happening? When the first filial generation or when the F1 plant produces flowers, etc., when gametogenesis takes place or gamete formation takes place, the gene for the tallness and the gene for the dwarfness, they don't get mixed. They get segregated without any mix. He called them as discrete bodies. They do not get blended, they do not get mixed, and they get segregated. And because of their non-mixing, because of their segregation, we are able to see the dwarf plant which did not appear in the first generation, its appearance in the second generation. So for this, Yeah, this is what we call law of segregation, otherwise known law of purity of gametes. Law of segregation, otherwise known as purity. What do you mean by purity of gametes? Here, what I mean to say is, though tall and dwarf are there, they are not getting mixed. Purity, they are segregating purely. Tall character is segregating and dwarf character is segregating in their pure form. So that's why we are able to see the character that got hidden in the first generation reappearing in the second generation. Now, he did not stop here. Then comes what we call, dihybrid. So dihybrid ratio here, what do you mean by dihybrid? So in the monohybrid, he has taken only one character at a time. In dihybrid, what happens? He has taken two characters. For example, round. The seed coat is round. Its opposite is wrinkled. And color, yellow. The color is green. So one parent was having seeds which are round and yellow. The other parent, wrinkled and green. And he crossed them. And in the first filial generation, like in the F1 or in the monohybrid ratio, the first filial generation, its progeny was exhibiting what we call round and yellow only. Wrinkled green got hidden. They did not express. So when the second generation was raised from the first generation, again here also, the round and yellow, wrinkled and green were there. In addition to this, round, green, wrinkled yellow. These two are non-parental combinations. We did not see them. They are the new combinations. So these non-parent and the new combinations we call recombinants. So these recombinants arose in the F2 generation. And when you speak of this phenotypic ratios, they were in the ratio of 9:3:3:1. They were in the ratio of 9:3:3:1 phenotypic. And when this F1 was crossed with double recessive, that is wrinkled green, and all organism or the gametes were produced in 1:1:1:1 ratio. That means the genotype are the round and the yellow. Though they are present in the one parent, round yellow are segregating independent of each other. Round character does not go with yellow. Round segregates on its own, yellow segregates on its own, wrinkled segregates independent of green. So like this, based on this, he proposed his second law or the third law as such, known as independent assortment. What do you mean by independent assortment? It's a type of segregation. It's a type of, generally we use the term segregation for monohybrid. When you take two genes at a time, or dihybrid ratio, this segregation we call law of independent assortment. So two genes are segregating or separating independent of each other. So this is what we call dihybrid ratio. And see, for all the 7 characters, he did not stop, taking a single character. He has done experiments, monohybrid, dihybrid experiments, taking 1 or 2 characters. For all the 7 pairs of characters he has chosen. And he got similar results were very, very similar. So he published all these findings, as I told you, in a journal. But they remained unnoticed for almost 35 years. He carried his experiments from, roughly from 1856 or 57 to 65 or so. But they remained unnoticed. Though they were published, they remained unnoticed. Nobody was interested. One reason was because, people like you and me, we are very poor in mathematics. And he was an excellent mathematician. He was very good at statistics. And generally biologists are very poor in mathematics. So that's why most of the people did not pay attention for his complicated work he has done and published. And it was in 1900. 3 people, De Vries, Correns, and Shermark, from 3 different countries. They have, what we call, they have repeated the experiments done by Mendel, and they too got same results. And this we call rediscovery of Mendel's laws. This work of De Vries, Shermark and Correns, we call rediscovery of Mendel's laws. It was done in 1900. Thereafter, number of people, attempted to repeat the experiments done by Mendel, and they did not get the similar results. Whatever the results he got, there were some deviations. Same result they did not get. But why Mendel got, uniform results, you'll be surprised. He was rather, he was lucky to say all the 7 characters he has chosen, they were present on 7 different pairs of, chromosomes. 7, they were present on 7 different chromosomes, all the characters he has chosen. So that's why segregation and independent assortment was without any deviation. But when other characters were taken into consideration, when the same was repeated in other, plants and animals, the people observed many deviations. And this we call, what we call deviations or aberrations. And this, if you, let us study one by other. One is in terms of monohybrid ratio. In terms of monohybrid ratio, he proposed what we call law of dominance. Law of dominance says that of the 2 characters, one character or one phenotype expresses itself in first filial generation, and the hidden character reappears in the second generation. So here what I mean to say is, one is completely dominant, only one character is observed. So one of the characters of the parents is observed in F1 in pure form. It resembles one of the parents in full, but deviation. Such deviation is what we call, incomplete dominance. Dominance, incomplete dominance means this was done in what we call 4 o'clock plant or snapdragon, Antirrhinum mazes, or 4 o'clock plant, what we call, Mirabilis jalapa. There, flowers, red flowers are there in one plant, and there are some varieties which give white flowers. When white flower plant and red flower plant were crossed, the F1 was exhibiting pink flowers. So it did not share the features of either of the parents. Parents were either red or white. But this new characteristic, what we call pink, came into existence. This we call incomplete dominance. This is incomplete dominance. While Mendel says complete dominance, but this was a deviation from Mendel. This is what we call incomplete dominance. There is one more, what we call co-dominance. Co-dominance. Here, see, tall character and dwarf character, only tall got expressed, and dwarfness got hidden, masked. So in another, experimentation, co-dominance. Co-dominance means it is observed, for example, there it is a common feature for you. Because your area, Ongole, is very famous for, Ongole bull. And then a cattle, if you take, there, they have done a number of experiments wherein he has crossed a cow and a bull. Cow, its skin was red and the bull was almost white. And the organism or the cattle or the or the young one, it had, skin, its skin color was in between. Red and white was there. Co-dominance. Another good example for co-dominance is, for example, we have blood groups. Blood groups are named based on the antigen that is present on the RBC. There are in all 3 major, or of course 4 blood groups are there: A, B, AB, and O. A means here A antigen is present. In B, you have B antigen. See, if our father is A, your mother is B, the children, you and me, have blood group of, according to Mendel, we should be either A or B. If A is dominant, our blood group should be A. If B is dominant, we should have B group. But to our surprise, we have AB group, not A, not B. We have AB. This means both A and B are codominant. Both are being expressed. This was a deviation from Mendel. Mendel said, no blending. They're discrete. But these are the variations for what we call Mendelian ratios. Now we move to the gene interaction, and which gives you the variations in terms of dihybrid ratio. Now here we know the what we call another, of course, let us take one more example also. Now, I told you, we discussed about what we call the incomplete dominance and codominance. Then we will also try to learn what we call another deviation, for example, lethal genes. Here, lethal genes, presence of a given gene, if a given lethal gene present in the gamete means that gamete or that organism shall not survive. See, for example, in some cases, in some plants, the chlorophyll, plants are autotrophs, animals are heterotrophs. The autotrophism is because of the presence of chlorophyll, and the chlorophyll is under the control of a gene. Let us suppose capital G is controls for chlorophyll, capital G. And in another case, for example, in another case, there is another, here G is dominant. Let us suppose G is dominant. If you cross G with a small g, and of course F1 will have, instead of GG, you have capital G and small g. And when this was selfed, generally we get 4 types of genotypes: GG, capital G small g, capital G small g, and then GG. And this GG, capital G, capital G small g, capital G is 1, capital G small g, they are 2, and small g, generally it should be, according to Mendelian ratio, it should be 1. 1:2, 2:1. 3:1 is the phenotypic ratio, 1:2, 2:1 is the genotypic ratio. Now here 1:2, 2:1. But the last one, small g, because capital G codes for chlorophyll. But when it got mutated into small g, it cannot code for, or it cannot produce the chain or the protein that gives rise to what we call chlorophyll. So as the chlorophyll is absent, the plant dies. So here, instead of 3:1, we get modification 3:0. That is, small g gets killed. So this is what we call lethal genes. If a given gene, either dominant or in the recessive form, if it is lethal, if it kills the parents or the offspring, then the ratio gets altered to what we call 3:0. Okay, so now we will learn about the variance in terms of or modifications in terms of dihybrid ratios. Now in dihybrid ratios, we have seen in dihybrid ratios, for example, we have a number of interactions are there, and these interactions shall alter the dihybrid ratio, dihybrid phenotypic ratio, that is 9:3:3:1 is the dihybrid ratio phenotypic ratio given by Mendel. Now this, this is possible when there is an independent assortment, but always this does not occur. And because the genes do interact, not only the genes that are present on the homologous chromosomes, but even the genes that are present on the non-homologous chromosomes are observed to interfere with the functioning of the other gene. So this we call what we call introgenic or interaction leads to the alteration of what we call phenotypic ratios given by Mendel. One such is, for example, complementary genes. This is complementary genes. Because of this complementary genes, the ratio, phenotypic ratio of 9:3:3:1 gets modified to what we call 9:7. Here, the presence of a single gene, see, both the genes control one character. Both the genes control one character. If those, for example, the white flowers. The white flowers are controlled by what we call, if capital C is present, flower is white. If capital P is present, flower is white. If capital P and capital C are present, there, the flower, new combination, that is blue flowers appear. Blue flowers appear. If only one single gene, one single dominant gene is there, either P or C, the flowers will be only single color. When both are there, that is white. When both C and P are together, instead of white, the flowers will be of blue color. So it is because of this, the ratio from 9:3:3:1 changes to what we call 9:7. So this interaction, what we call complementary, they are complementary. The gene for color, that is C, and the gene, another gene for the color, that is P, both are complementary to each other. They help each other. Even this, either the presence of capital C or the presence of capital B is sufficient to give white color to the flower. But when both of them are present, instead of white, they give what we call blue coloration. So here, these two genes are known as the two genes that are mutually helping each other. They are known as complementary genes. So it is because of this complementary action of the genes, the ratio gets altered to what we call 9:7. Then the other is what we call epistasis. This is another modification. Epistasis means if there are two genes, if one gene masks the expression of the other gene, the gene that masking is called epistatic, and the gene that got masked is known as hypostatic. So this epistatic gene and hypostatic gene interaction, it is of two kinds. One is dominant epistasis, the other is recessive epistasis. So epistasis means one gene masking or suppressing the expression of other gene. The gene that is suppressing is known as epistatic. The gene that got suppressed is known as hypostatic. So this epistatism may be by a dominant gene or by a recessive gene. If it is by a dominant gene, generally the phenotypic ratio gets altered to what we call 12:3:1. 12:3:1. This was observed in what we call in a squash pumpkin. So in the pumpkin, the fruits are of fruit color. There are three kinds. Some white, some are yellow, some are green. The gene for white, it is epistatic over both, that is yellow and green. So that's why if a single W is present, means all such combinations are white only. And the second one, that is yellow, capital Y. This capital Y is dominant over green. that is small y. So when W is absent, when capital Y is there, small w is there, the fruit's color will be yellow. And the green color will be there in the fruit only when small w and small y are present. So this is what we call dominant epistasis. And the other is what we call recessive epistasis. This was observed in mice. In mice, the hair of the mice, there are three kinds. In some, the black color hairs are there. In some other, agouti. And the last one is what we call albino. The hair is white. So in, of course, black color, I need not explain. Agouti means here, the color, hair color is black, but here and there you find yellow patches. That is what we call agouti. Albino means white. So here, it is because of the recessive epistasis, the Mendelian ratio of 9:3:3:1 gets altered to what we call 9:3:4. This what we call recessive epistasis. There is one more another kind what we call duplicating genes. Duplicating genes here, in contrast to the other characters which we have just studied, here the character is controlled by genes that are not present on the homologous chromosome, that are present on different chromosomes. Say, for example, this was observed in what we call a weed. A weed generally that is present, it is what we call shepherd's purse. Shepherd's purse, a weed, it produces fruits. The fruits are of either triangular or top-shaped. Triangular top-shaped fruits are there. In shepherd's purse, a weed, two types of fruits are there. Some are triangular, other are what we call cylindrical or roughly top-shaped. Now here, both the genes, that is T1, T2, T2, both the genes determine the shape of the fruit. In all cases, whether capital T1 is there or capital T2 is there, just presence of one dominant gene is sufficient to give a particular phenotype. So in all cases, in all the 15 cases, you find only triangular fruit, but only one case, that is small t1, small t2. In this combination, double recessive offspring, you find the presence of production of the fruits that are top shape. This we call what we call duplicating gene effect. Duplicating gene effects. So these are some of the deviations. I told you already. So deviations are because of the interaction between genes. But he did not visualize. Mendel did not visualize such deviations because he was lucky. All the characters he chose were present on different chromosomes and free independent assortment was there. But here, a new phenomenon, what we call, these are because of what we call presence of different genes on the same chromosome, and they are present very close to each other. This phenomenon, what we call linkage. It is because of the phenomenon of linkage, the parental combinations are more in number and the variants or the recombinants are in less number. But according to Mendel, they should be present in 1:1:1:1:1. All what we call when you cross a F1 parent with a double recessive parent, F1 individual, when it's crossed with a double recessive parent, you get all the gametes in equal numbers. So they are present in what we call, they get segregated into what 1:1:1:1:1. In normal cases, these were the results which were seen by Mendel. Now, we move to what we call deviations, the reasons for these deviations. the reasons for these deviations, for this we study two important phenomenon, what we call, linkage and crossing over. Linkage and crossing over. What do you mean by linkage? Here, I told you the genes are present on the chromosome. The genes are present on the chromosome in the form of what we call beads and a string. Like beads and a string. If you take a thread and if you, put beads one after the other. So like this, the genes are present on the chromosome, or more specifically your DNA, or a part of DNA. Of course, while studying DNA, I told you nucleosomes, wherein, chromosome or the DNA material, it makes two rounds. It winds around histone protein, and that structure, that results what we call nucleosome. So it is because of this nucleosome, it is because of this twisting, or it is because of this supercoiling, large or a big molecule of DNA got condensed into a small microscopic chromosome. So now let us study about the phenomenon of linkage and crossing over. Now this linkage and crossing over, this theory of linkage was given by Betson and Punnett. Betson and Punnett, while they're working on possum species, they observed that according to Mendelian ratio, when the F1 is crossed with a double recessive, the gamete should be 1:1:1 ratio. But that was not the case. In the experiments done by Betson and Punnett, they observed that the genetical or the parental combinations were in large number, whereas the recombinants were in less number. The recombinants were in less number, whereas the parents number or parents types outnumbered the recombinants. Say, for example, there are, see, for example, they have crossed, they have taken two, plants, what we call, purple color, and pollen was long. Purple with long pollen, and the other was red flower with round pollen. The purple is dominant over red, and the long pollen is dominant over a round pollen grain. So when they were crossed, the F1 was, of course, the F1 was with purple flower and long pollen. When this was, test cross means what? When F1 is crossed with double recessive parent, that cross we call a test cross. To know the genetic constituent of the F1, we resort to what we call test crossing, wherein F1 is crossed with a double recessive parent. Now here, when this was done, when test cross progeny, purple and long were 192, and the red and round, this is another parent, this was 182, whereas the crosses, that is purple round, originally purple long, but purple round and the red long, these are recombinants. These recombinants were just 23 and 30. So this shows us that the number of the parental organisms that resemble parent are outnumbering the recombinants. How this is possible? This should not be the case. Whereas Mendel said all will segregate independent of each other and they are produced in equal numbers. But here it is not the case. Parental combinations are outnumbering. They are produced in large numbers when compared to the recombinants or non-parental combinations. Some organisms that are produced or some individuals that are produced, they are differing from both the parents. They are known as recombinants. So recombinants are in few number, but parental combinations are more in number. Now why this is? He termed, this phenomenon of existence, coexistence or close existence of certain genes and the entry of these closely packed genes into the organisms together. Passing to together is what we call, phenomenon of linkage. So a mechanism by which a group of genes and a particular chromosome show what we call tendency to link together, to remain together, is what we call linkage. It's a phenomenon that, wherein the closely or the genes that are present on a given chromosome, when they are closely linked, they tend to move together. They tend to move together. They do not get segregated. They do not get separated. So this tendency of moving of closely linked genes together is what we call linkage. It is because of this linkage, parental combinations are produced in large numbers. So here they are in very, very large numbers. They are present in very, very large numbers. And this what we call, phenomenon of linkage. This, what we call a linkage. See, in another example, for example, this was done by Hutchinson in maize. Same linkage. For example, in Hutchinson, Hutchinson did his experiments on maize. In maize, he's taken, a plant with endosperm which is full, and seeds are colored seed with full endosperm. The other contrast one is wrinkled seed with a wrinkled endosperm and the colorless seed. When these two are crossed, generally the F1 will be colored and, colored and with full endosperm. Colored full. And when this is test crossed with the parent, that is wrinkled and white or colorless, and the gametes that were produced, they were, for example, seeds with full seeds and colored, they were produced in 4032 numbers. And the other, similarly, the other parent, that is wrinkled and colorless, they were produced some 4035. And the recombinants, endosperm with full endosperm but colorless. The other is wrinkled endosperm, colored. These are the new recombinants. They were produced in 150 and 149. They were. So the parental combinations are in roughly 4,000, whereas these recombinants are in just hundreds. So by doing mathematical calculations, we can come to a conclusion that the genes that control the shape of the endosperm and the color of the seed, they are closely placed. They're very close. So, and if they're far apart, if they are distantly placed, then in such a case, you will see both parental combinations and the recombinants almost in equal number. Higher the space, generally more recombinants are. If they're closely placed, very tightly packed, in that cases, the parental combinations are more, whereas recombinants are less in number. So this tendency of moving of the closely linked genes together into a gamete is called what we call a linkage. Now, so not only this, even in human beings also, we have a number of genes which are linked. Say, for example, most of the genes that are linked, you find in sex chromosomes. The genes that are present on the X chromosome are known as X-linked or sex-linked. X-linked or sex-linked. And of course, even there are certain genes which are linked to Y chromosome. So they are known as Hollandric genes. X-linked genes, Hollandric genes. Y-linked genes are otherwise known as Hollandric genes. Of course, the X-linked genes, because they are linked to X chromosome, they do not move to Y chromosome. So certain genes are, certain characters, pass from mother to daughter and son equally. But whereas from a man, the characters that are Y-linked, Y chromosome moves only from father to the son, not to the daughter. But from mother, you get an X. From father, you get a Y. Daughter gets X from mother, X from father. XX. But son, XY. So if Y-linked genes are there, means they are they are man specific or male specific. They get transmitted from father to son only. But if it is a X-linked, they move either to the son or to the daughter. So, for example, good example, hemophilia. The clotting of blood doesn't take place. It is a sex-linked. Hemophilia. And the other is, for example, color blindness, wherein you can't see green and red. This is very dangerous, Generally, if you're colorblind, generally, they don't give you license to drive. If there is you don't know, you cannot distinguish whether it is green or red light glowing. So this is what we call color blindness. Color blindness is another X-linked gene. And a good example for Y-linked gene is what we call Hollandric. Generally in males, generally on the what we call on the ears and the pinna, you will see long hair. This is Y-linked. This is what we call a Y-linked gene, which is Y-specific or male-specific. So these are linkage. This is what we call linkage. So if linkage is there, through linkage, what do you understand? Here means chromosomes, on the chromosomes, genes are very closely packed, closely placed. The strength of the linkage depends on the closeness. If they're very tightly packed, closely placed, generally recombinants will be very, very less. If they're distantly placed, generally recombinants will be more. Now, then we move to what we call crossing over. Crossing over. Yesterday we learned about it. While learning about the process of meiosis, I told you in the meiosis 1, in the prophase, we have seen for our convenience, we divided that into 5 subphases. Leptotin, zygotin, pachytin, diplotin, and diakinesis. In leptotin, what happened? The condensation of the chromosomes, the chromatin takes place, and you will see, chromosomes, homologous chromosomes coming together. When it enters into zygotin, generally, the synaptonymal complexes and bivalents are formed. When it moves to what we call pachytin, there you will see each chromosome, each homologous chromosome splits into 2, and you will see what we call pachytin tetrad. You have seen, I told you. In pachytin tetrad, what happens? The 2 non-sister chromatids, they become like this. They become like this, and you will see X-shaped structures. This we call chiasmata, and these areas represent, a point where the chromatid got broken and where the exchange of chromatids has taken place. And it is because of that, the recombinants, it leads to what we call formation or creation of recombinants. Now, the crossing over means it always takes place between non-sister chromatids. When they come together in pachytin tetrad, and these 2 non-sister chromatids, they move around each other, they twine each other, and they cross with each other. And there are 2 theories, to express the phenomenon of crossing over. One is what we call traditional theory, and the other is chiasma theory. And according to this traditional theory, and this theory, it was given by Sharp, L.W. Sharp, who says that during pachytin tetrad, generally they say that it is because of this twisting or overlapping with each other, a strain will develop. The tetrads will, or the non-sister chromatids, develop a kind of strain, and at these strains, because of this strain, the strain happens at the chiasma places. So chiasma are formed first, and at the chiasma places, the chromatid breaks and the exchange of chromatids takes place. So according to this theory, chiasmas are formed first, then the exchange takes place. But reverse is the case for chiasma theory. This was given by what we call Belling and Darlington. They say that, first of all, breakage takes place. Later on, exchange takes place. And the chiasma represents the areas where the exchange has already taken place. So breaking first, then the exchange. Whereas in a traditional theory, there they say that first of all chiasma are formed, then the exchange takes place. So whatever the case may be, whatever the case may be, the exchange of the chromatid pieces takes place between the non-sister chromatids, and this leads to what we call, and we have seen, then the repulsion takes place, then the termination takes place, then the homologous chromosomes move to different poles, we have seen. So this results in what we call this exchange of chromatid pieces among the non-sister chromatids is what we call crossing over. This is what we call crossing over. It is so the frequency of crossing over depends on the distance between the genes that are present on the chromosome. If they are closely packed, the frequency of crossing over will be very, very less because closely packed, no scope. If they are distantly placed, easy exchange takes place, crossing over takes place. So the crossing over is determined by a number of the number of things are there that determine the rate of crossing over. So the crossing over is influenced mostly. The crossing over is influenced by a number of features, environmental features, and even chemical features, chemical substances, and radiation, ionizing radiations, etc. So many extrinsic and intrinsic factors have been found to affect the rate of crossing over. They are first, age. It depends on the rate of crossing over, depends on what we call age. In old age generally the rate of crossing over will be less, but the strength of linkage will be more. So as the age progresses, linkage will be more and rate of crossing over will be less. Coming to the temperature, if you increase the temperature, environmental temperature increases, then the rate of crossing over increases. If the cell or the organism is exposed to ionizing radiations, such as X-rays and other ionizing radiations, they help in enhancing the rate of crossing over. And chemical substances such as ethyl methyl sulfate, EMS, etc., they enhance the rate of crossing over. So this is what we call crossing over. And with these frequency of crossing overs, we can construct what we call chromosomal maps. What are chromosomal maps? A chromosomal map will give you how many linkage groups are there. Generally, the number of linkage groups of a given species equivalent to its n number, haploid set. Say, for example, our haploid set is 23 or 23 pairs. We have 46 chromosomes, 23 from father, 23 from mother. So n is 23. So the maximum number of linkage groups a human being has will be roughly 23. If it is Pisum sativum, 14, 7 pairs. So the linkage group will be determined. You get the idea of linkage, the number of linkage groups, and their sequence by doing what we call 2-point test cross and 3-point test cross. By performing these test crosses, you will know the position of different genes. Then the third is the distance between two genes will be determined by the percentage or the frequency of the crossing over. Higher the crossing over rate, generally more will be the distance. Closely packed, less crossing over percentage, less number of recombinants will be there. So this is, so based on the linkage percentage or frequency of the crossing over percentage, we can construct what we call chromosomal maps, which will give you the number of linkage groups that are there in that individual and the number of sequence of chromosome, the genes that are located, and the distance between two genes will be determined by the frequency or the percentage of crossing over. This is what we call chromosomal maps. So this is about what we call. So today we learned about what is Mendelism and what is what is dihybrid, monohybrid ratios, and what is phenotype, what is genotype, etc. We have seen, and the reasons for the success of Mendelism we have seen, and the rediscovery of Mendel's laws we have seen, and the deviations, that is incomplete dominance we have seen, and codominance we have seen, and also deviations in dihybrid causes we have seen, wherein we have seen what we call complementary genes, epistasis, the dominant epistasis, recessive epistasis, and etc. We have, and the lethal genes, etc. we have seen. It is because of this interaction between genes, the phenotypic ratio given by Mendel gets altered. This we call gene interaction or modification of the phenotypic ratios given by Mendel. And we have also learned about what is the meaning of linkage, what is crossing over, and the theories that explain the phenomenon of crossing over we have seen, and the chromosomal maps we learned a little bit, of course, and the linkage groups and the positioning of various genes and the distance between individual genes, etc. we have learned. So this is about the Mendelism and the modification of interaction of various genes, and that leading to what we call modification of Mendel's ratios. So okay, Navya, any doubt, Amma? Hello?
Yes, sir.
ఏమైనా డౌట్స్ ఉంటే అడగమ్మా.
ఏం లేదు సార్.
ఎందుకంటే ఇక మనం కొద్దిగా ఫాస్ట్ గానే పోతున్నాము. ఏమి లేదు, నువ్వు ఇంతకుముందు నిన్న కూడా చెప్పాను, ఈ ఇంటర్మీడియట్ లో చదివినటువంటి బుక్స్ దగ్గర పెట్టుకొని ఒకసారి అవి చదువు. అవి చదివిన తర్వాత ఇందులో ఉండేటటువంటి ఇంకా ఉన్న కొద్దిగా అంటే లింకేజ్, క్రాసింగ్ ఓవర్ అనేది కొద్దిగా అంత ఎక్కువగా లేదు. డీవియేషన్, జీన్ ఇంటరాక్షన్ అనేటటువంటిది కూడా ఎక్కువగా, మోడిఫికేషన్ ఆఫ్ మెండల్స్ లాస్ కూడా ఎక్కువ అందులో లేదు. అవి అలాంటివి కొన్ని చిన్నవి ఇందులో యాడ్ చేసుకుని, ముఖ్యంగా 10th ఇంటర్మీడియట్ ది బాటనీ ఇంటర్మీడియట్ బుక్ కొద్దిగా ఎక్కువగా చదువుకుంటే నువ్వు ఎగ్జామినేషన్ చాలా కాన్ఫిడెంట్ గా రాయగలుగుతావు. సో మనం నెక్స్ట్ ఇంకొక 2 క్లాసులు ఉన్నాయి మనకు. సో we will be meeting and discussing about the rest of the syllabus. That is mostly we will discuss about what we call a genetic code and protein synthesis in the next class. Then of course genetic engineering, etc. in the last class. Okay, so see you in the next class, that is on afternoon of next Saturday.
Thank you.
Okay, Amma, thank you.